Hello, everyone. It's my pleasure to be here today to present this paper called the Raman Photothermal Microscopy. The paper will appear in Science Advance in a few days. With this, I would say that this paper is a jump from the earlier pioneer work by the photothermal people. So it's my pleasure to be here today to report this to the photothermal community, also to the chemical imaging community. So please allow me to start with a big picture introduction of chemical microscopy. We know that the optical microscopy is a foundational tool for life science, especially with phase contrast. We can visualize the intracellular structures, but they don't provide the molecular information. To visualize biomolecules, we use fluorescent microscopy, and especially the discovery of these green fluorescent proteins, which allow us to visualize protein dynamics and the function of cells, like neuron cells. So these are great, but this does not answer all the questions. We still can not visualize many molecules, especially small molecules. There's still a big gap here, right? Just give an example of cholesterol. This is a very important molecule in our body and in our cells, but we still have no means to visualize cholesterol trafficking and metabolism inside a single living cell, because any molecule will be much bigger than the cholesterol perturbates function. An elegant way to solve this problem is using the chemical bond vibration. So in this case, is this double bond in cholesterol, which give a fingerprint of this molecule. Also, the chemical bond is very small in size. It's about 1 angstrom, which is much smaller than any molecule. So we can using specific chemical bond to tag as a GPS of molecules with minimal perturbation. So this concept has been very attractive for many years, but it's technically very challenging because the signal from a chemical bond vibration is much, much weaker than fluorescence, and innovations are needed to overcome this huge barrier. Over the past 20 years, advanced chemical microscopy has been developed. So here, the chemical bond vibration can be measured in 2 ways, just a background. One is spontaneous Raman scattering, and the other infrared absorption. One measures the molecular polarizability, and the other measures the dipole moment. However, they have difficulties. The Raman scattering suffers from a very small the cross section, which makes the speed very slow, and the infrared has a much bigger cross section, but it suffers from a very poor spatial resolution and also the strong water absorption. Over the past 20 years, coherent Raman scattering microscopy has been developed. Instead of using 1 beam in spontaneous Raman, we use 2 beams, one called the pump and the other called Stokes. And when the energy difference tuned to drive a chemical bond vibration, this produces a strong signal called cars at a new frequency, or you can measure the stimulated Raman scattering or in intensity gain or intensity loss in one or the other beam. This can be done in 2 ways, narrow band or broadband with a femtosecond laser. So CARS and SRS has become a mature field with commercial products available. In 2012, we have the first book called Coherent Raman Scattering Microscopy, and in 2022, we have published the second book called the Stimulated Raman Scattering Microscopy by many experts of the field, and you can read these books. So with this, I'm going to say that every technique has its own limitation. Back in 2015, when working on this review article, we realized that the detection sensitivity of CARS SRS is about 1 millimole for endogenous molecules or single chemical bonds. And that's not sufficient because many molecules in the cell, like drug molecules, specific nutrients, their concentration is at the minimal, micromole or nanomole level. So we need to push the sensitivity of the field to be compatible or comparable to fluorescent microscopy. And this has been a huge. So in the following, I will basically tell you a 16-year story that how that led to this stimulated Raman photothermal microscopy paper. Back in 2007, that's 16 years ago, my group was studying the photo damaging CARS microscopy. And as a co-inventor of CARS, my original plan is to tell readers that there's no photo damage in CARS microscopy, everything is perfect. But the observation is exactly the opposite. We actually see an enhanced photo damage at the 2840, which is CH bond. we did the theory and showed that it is due to not due to cars, it's actually due to the stimulated Raman gain and the stimulated Raman loss that cause the energy transfer, and this thermal toxicity that increase further enhance the damage if you do point scan here. So this is, I was thinking the other way that because this energy deposition, we can do something, right? We can do something with such an energy transfer. So at that time, I was at Purdue University. We spent 2 years building this stimulated Raman photoacoustic microscope. My colleague Bob Lacht in mechanical engineering was a pioneer. He is still a pioneer of CARS microscopy, and he has a nanosecond OPO that idled for many years. So we brought Olympus microscope into mechanical engineering and built the system. We worked for 2 years, and we never got signal because it turns out the transducer was not sensitive enough to measure this stimulated Raman effect. Meanwhile, the optical SRS microscopy has been published and has become a big success. But the student working on this project, Han Wei, he's a very hard worker and very persistent. And by accident, he found a strong signal, photoacoustic signal from an overtone vibration. And this overtone vibration is also due to chemical bond vibration. And this opened a new direction of vibrational photoacoustic imaging, and has allowed us to visualize this in lipid and cholesterol from the Newman side, exciting from Newman side in cardiovascular disease. We continued along this direction, and recently we're using photoacoustic to do neuromodulation. But meanwhile, the efforts of pushing the detection limit in chemical imaging continues, right? So this time by thinking out of Raman box. So as I mentioned earlier, that the Raman suffers from a very small cross section, and then outside the box we had the mid-infrared absorption, which have a much bigger cross section. And the calculation here, this is a DFT calculation, shows that for certain group like azide, which is used in clinical chemistry, has a cross section 10⁻16. That is huge compared to Raman, and this is at the same level of electronic resonance, which produce fluorescence. However, unlike electronic resonance, IR absorption or vibrational absorption, they don't produce any emission, but it produces heat. So now you see that the photothermal detection become a natural means of measure chemical bond. Right? So I presented this paper 2 years ago in the first photothermal webinar. So this is called a mid-infrared photothermal microscope. The idea is that we can use a pulse IR to pump the sample and using visible probing. And by using so this change the local refracting index or thermal expansion of a particle. By visible probing the thermal effect, we can reach the visible diffraction limit. So this microscope for the first time that allow us to see the chemical bond in living cell, the membrane, and lipid. So the microscope is very compact, was built by Delon, who is now a professor at Zhejiang University. So the excitation is a QCL, high compact QCL laser. The visible is a CW laser. So it's also that the cost is much lower than a CARS microscope. So back in 2016, October, this is 1 month after I published the paper, I give a talk in SciEx meeting. And I was very lucky because there are 2 important people in the audience. They are the CEO of Anesys, Roshan, and Anesys was a world leader of AFMIR. But a big problem, AFMIR, although it can allow nanoscale imaging, cannot probe molecule inside cells. So that's essential for life science study. And they see the paper and they are very excited. They said they want to convert this into a product. And only in 12 months, they announced Mirage, which means the contrast from refracting index change. And in 2018, this photothermal spectroscopy is formed, and now Mirage has delivered to many labs and produced many publications that is beyond my own imagination, because we are still focusing on life science. Also in 2017, I moved from Purdue to Boston University, and we have developed this technique in 3 ways to give this community update. The first one is that we do confocal MIP, in which we scan the beam, and recently we have reached the video read. The second method that we do camera-based MIP microscopy. So this is like a parallel detection, so they can match the visible and IR focus. So you can do high throughput study till the frame rate of a camera. For example, appear 1,000 frames per second chemical imaging speed. The third method is a collaboration with my colleague at the Boston University, Lei Tian, and they're using the chemical computation approach to produce a 3D map of refractive index called a phase contrast tomography. And we use IR to introduce a modulation of this 3D map of refractive index. We can do volumetric chemical imaging. So I will not go through the details, but just show you one example of this video read imaging built by Jia in the group. So the idea is you synchronize scan the IR and the pump, and you do single pulse digitization of single pulse induced photothermal signal at each pixel. So you can do video read imaging. This is significant because this means that when we do this imaging, when we do video read imaging, we can continue to tune the QCL laser. So now at each pixel, we can produce the IR spectrum at a visible diffraction limit of 200 to 300 nanometer spatial resolution. So this allows functional imaging. For example, we can now decompose a single cell fungi wall 100 nanometer into 3 layers because they have different composition. We can also imagine the state of a protein, for example, or imaging a chemical reaction in living cells. I will not go into detail. But with this advance in the lab, I never thought to using Raman or CARS, SRS to photothermal imaging because I was brainwashed, right? And because of the earlier failure. So the Raman and SRS is too weak to do this kind of experiment. And the situation changed in 2021, which we had a party, and this is for my 50th birthday. A major theme of the party is sports, and we play, students play basketball here. And you can figure out that one of the students here in this picture, he's got injured, and the doctor told him that you cannot walk for 2 months, and that was a nightmare. And but there's a Chinese saying that the bad thing can become a good thing, and we said you cannot do anything, so you probably can do a calculation for us to calculate this stimulated Raman photothermal effect, because we need to figure out that whether thermal toxicity or nonlinear toxicity SRS imaging. So for many years we do SRS, we always do stimulated Raman gain and the stimulated Raman loss, and we think that we did everything because that's everything we have, right? But we paid a little attention to what happened to the sample. SRS is essentially a 2-photon vibrational excitation, pump and dump. And then this decay produces a heat that can produce a photothermal effect. So since the Yifang cannot do any experiment, so he did a calculation of this using our earlier paper in 2007 and a more recent paper of this multiplex SRS. Basically, if you know the modulation depth, you can calculate the energy transfer theoretically, and there's a simple equation here. So he did a calculation, shows that now with 25 milliwatt pump and 50 milliwatt Stokes on the sample, this is the power we use routinely for SRS imaging, and there's an energy transfer of 2.5 microwatt. And if you use a single pair pump and Stokes power, that'll produce 31.3 femtojoule. That sounds a very, very small energy, but into a very small femtonator space, right? So he Yifang further built this diffusion model based on Newton's law of heating and cooling, and this is a diffusion model. And with this model, the result is quite surprising. With 10 microsecond pump and Stokes powers at this 25 milliwatt and 50 milliwatt Stokes, in less than 10 microsecond, you can reach the DMSO temperature by 2.4 degrees. So how to prove this? At that time, we turn to fluorescence, because fluorescent dye, like rhodamine B, are very sensitive to temperature. It's about 1 degree per 1% intensity loss. So we put a rhodamine B into DMSO solution. We do SRS excitation, and we do 2-photon fluorescence same time. And by monitoring the 2-photon fluorescence, we see that there's an intensity drop of a few percent in 3% in less than 10 microseconds. As a control, we use rhodamine 800, which is non-thermal, and there's no change over time. So this indeed shows the loss. So overall, there's a few percent change a few degree temperature increase, there is a slight mismatch because one is SRS modeling and the other two-photon fluorescence, which is the focus size a little bit different, but the concept is exactly the same. So this is huge because based on my MIP study, we know that this can produce a substantial photothermal signal. And this signal is actually consistent with my friend Wei Ming. He did a very nice theory work recently. He modeled SRS as a two-photon absorption process and with a cross-section similar to electronic two-photon absorption. And if you consider high the Stokes power, you can calculate apparent sigma cross-section, which essentially the modulation depth. And this cross-section is a little bit lower than infrared, but much higher than Raman. So this is very consistent with our observation and said that this energy transfer can be substantial. So in our lab, we spent since that when Yifang was recovered and went back to lab, and he and Xiaowei together built a stimulated Raman photothermal microscope to measure this stimulated Raman photothermal length, right? It's based on SRS, but it's different from SRS. So here we do a modulation both pump and Stokes, and then we can use a third beam to produce a probe. And this third beam can be you can just do second harmonic from the Stokes, so you don't need an additional laser beam to do this experiment. So you do this, and then you measure the SRP signal. You can do single pulse digitization because the thermal process is on the microsecond scale. We also built a ball-length model to model this new type of microscopy. It contains three terms. The first term here, N more, that is the number of SRS event. The second term, H bar omega SRS, that's the energy per photon. So the first term times the second term is the total energy deposition. The third term is tell you how this energy convert to refracting index change, the thermal length. So that's the theory of SRS. So it's a linear to the concentration, it's a linear to the pump and Stokes energy, but also depends on the dNdt. We also can use in low duty cycle. So this is same for SRS. So using low duty cycle, you can actually enhance the peak power so that you can increase the number of SRS event to further increase signal. So with this, we apply this to the SRP to DMSO, and the results is very surprising. We actually can see the signal by eye because now the modulation depth is by 22.3%. And this is induced by the photothermal lengthening effect. And this is tremendously bigger than 0.05% in the best we can do for SRS measurement. Also, you can see that here that we can digitize this photothermal process to photothermal dynamic imaging, and this we see the fast decay, and you can convert it into frequency domain, and you can see high harmonic signal. You can put a match filtering to remove the noise and go back, and now we can get a very strong signal, and this signal is from a 100 nanometer particle. This was very difficult to do if we do CARS or SRS microscopy. So we also measure liquid, and this is for CH bond. We have 2.3 versus 39 minimal limit of detection for CH bond, and for CC triple bond we have 2.1 minimal detection versus SRS 26 minimal under the same pump and Stokes power. So we are about 10 times better detection limit. This is you can apply this to living cells. So this is living cancer cells. You can see the lipid dynamics, and also the see other organelles that are more rich in protein. In this case, you can apply this to brain slice. Here, you can see the minor sheath in deeper brain or white matter, and you can see this is in the gray matter. You can see the intracellular protein signal. You can also see this the CD signal by this fatty acid treated cancer cell. And the offering, you do see a contrast. And this is a trade-off for SRP microscopy, because when we do photothermal, each of the pump or Stokes beam can induce a linear absorption by overtone or two photon. But this linear absorption can be removed by advanced modulation, such as delay modulation or spectral modulation. Key aspect of photothermal microscopy as the theory done by Michel Orid and earlier pioneers of the field is that the signal depends on the heat capacity of the solvent. So if we use a glycerol here, like a tissue ammonia medium, it has a smaller heat capacity and also smaller thermal conductivity than water. So this can further boost the signal by about 10 times. So if we merge this glycerol into PMMB particle, and now you can see a very strong SRP signal from single particles with 200 nanometer resolution. And in SRS, you would only see noise. If you apply this to cells and now compare SRS and SRP, we can see that the SRP will give a much better signal to noise ratio in the single cell level. But this method is most useful for doing tissue imaging because glycerol is a natural tissue clearing agent, and you probably can apply other ones. So you can now do fingerprint imaging of this amino-1 band lipid CC double bond with very high signal noise ratio. If you do SRS, it will be very noisy in this fingerprint window. So that's an advantage of this for tissue imaging. But now, please allow me to give you a few seconds to compare these two modalities. Certainly that a trade-off between them. So SRS measures the total amount of photons. So you need a high NL objective to collect all the photons. Otherwise, you will see a huge cross-phase modulation actually induced by the photothermal effect, right? But the SRP exactly measures the photothermal effect. And we actually need a pinhole to reduce. And so we can naturally use the air condenser to measure the SRP signal without any degradation of the image quantity. So with the air condenser, you can get a beautiful SRP image from these particles. But with SRS, the quantity is much reduced. And you can see that the signal noise ratio is 10 times better if you do SRP measurement. From the customer point of view, this is very essential because the biologist would just put a dish there and do forward SRP and the backward confocal fluorescence microscopy without another like oil objective to do this measurement. Another thing is the laser noise. In SRS, directly measure either pump or Stokes, depends on Raman gain, Raman loss. Right? So it strongly depends on ring noise in the pump or Stokes beam. SRP is less sensitive to the excitation laser noise because we use a third beam to measure as a probe. So the consequence of doing this is that you can use a compact laser. I bought a refined laser OPL to do SRS measurement. And to do that, we have built a very high quality balance detector to get a good image. But with SRP, you can get a beautiful images without a balance detection. This was the data last year. Another one is I mentioned that I actually want to make this into a product because this actually you can do all this measurement with the air condenser that bring convenience to the life science community. So this is a newer image we got recently with SRP without a balance detector. You can get a beautiful image from a cell, right? And if we do SRS with a balance detection or without a balance detection, that the signal noise ratio is worse than SRP detection. Finally, that one more thing I think is really the future of the field is that you can do single pulse SRP with a low pulse. This low frequency, high energy laser. You all remember this 3-photon fluorescence by Chris Xu, and that creates a big community of doing. And that need a high energy laser. But this kind of laser cannot be directly used to do SRS microscopy. We actually bought this laser from Newport, but the pulse to pulse fluctuation of 1 to 2%. So you usually cannot do too much. But because SRP is an indirect measurement, you actually use a third beam to measure this, and we can get a beautiful signal. This now in the oscilloscope, you can see this photothermal signal is spike. These are induced by individual pulse, one pulse, high energy, not a microjoule pulse, and in the spectral domain, you can get beautiful signal, and you can get like up to 3,000 or higher signal noise ratio just by single pulse produce SRP signal. So we are still working on it. We hope that this is probably the solution to really push the sensitivity from millimolar to micromolar level and to see low concentration molecules. And that is really the hope that we can do something beyond the reach of fluorescence microscopy. With this, I'm happy to summarize. So today I mentioned that this, I call it VIP, vibrational photothermal microscopy. I present two modalities to you. The first modality is I present in the past photothermal webinar. It's called a MIP. So MIP using the IR excitation called local change of effect index and the thermal expansion, and we use a visible beam you show here to probe the photothermal effect and induce a visible diffraction limit. So number one. Number two, because much bigger IR cross section, we can reach micromolar detection sensitivity. I don't have time to show you here, but this applies for CO bond, for protein, CN bond, nitrile group, and azyl group, and we can routinely achieve micromolar detection sensitivity. The second method I show you today is we realize that SRS is essentially a two-photon vibrational absorption process. It could heat up the sample at a few Kelvin level. So by this two-photon vibrational absorption and the thermal decay, we realize that this stimulated Raman photothermal effect can produce a high modulation depth of 22% for pure DMSO, right? And this pave a way for high sensitivity chemical imaging at the ease of operation, because this can be done with air condenser with a simple. Actually, my students show this, you can do this with a $100 lens for signal collection. So it opens enormous opportunities and could together help, or together with SRS, could further boost the broader use of this chemical imaging tool. So with this, I will stop here and thank you for your attention. And I'm happy to answer questions.
Thank you, Professor Cheng. It was a really great talk, and quite amazed to see how many developments is happening in this vibrational spectroscopy, and especially I was quite amazed with this 22% modulation depth in the SRP. So that's a really great achievement. So we are now open for questions.
And I got a
so
question. I got a question from the pool, from Shuhua, my former student, who is now in Beijing. So she asked that how fast can it achieve? Yeah, that's a good question. So what we can, I believe that with this NOPA, which is 1 megahertz, right? So you can do basically 1 pixel, 1 pulse per pixel. So we can reach a video rate if we do 1 pulse per pixel, and that is the speed, and that allow us to hyperspectral imaging. And how large a field of view? This is similar to SRS. If the field of view is similar to SRS, and if you do millimeter scale, you can stitch the sample scan, the laser scan together to get this. The second question is from Marcus. Do you have experience of sample degradation? With sample degradation when doing SRS? I'm not sure what do you mean by sample degradation? You mean the damage to the sample?
I think Marcus, you can speak it.
Okay, thank you. I think you can hear me.
Yeah,
yes.
I can hear you.
So now you can see me. Hello. The main thing is that the high intensity you use for Raman scattering may affect the sample in contrast to the low intensity you use for normal IR absorption. And that is why I was wondering whether just sending IR light and let the IR light absorb affects the sample less than sending in high energy visible light with high energetic photons.
I totally agree. Yeah, because SI is using the same beam for 2 photons, so it will cause photo bleaching, for example, right? In that sense, IR is safer because IR only purely produce heat, nothing else. So that you're right. Yeah. And that's one of the disadvantages of SRP compared to MIP I earlier developed, because MIP cannot produce any other background, but SRP also, each beam can produce a linear absorption, and that's but SRI has its own advantage. it's have no water absorption, right? It can go into the tissue. Yeah. So, yeah. Okay.
Okay, thank you.
No problem. Any other questions?
Is there other questions? You can speak it up as well.
Yeah, you can put in the chat as well.
Yeah, you can also write in the chat.
I just want to mention that you can, if you're interested, you can do this experiment in several ways. One is that if you have a fiber laser, you can do it immediately because this is you don't need a band detection to do this. And second, if you use the inside, you don't need to buy any additional one because you can just simply double that the 1040 beam as a probe, and so you have all the laser source to do this measurement.
I would have an additional question, maybe for the IR version of that. You say that the detection limit is micromolar detection sensitivity, or so yeah. What is really limiting the sensitivity at the end?
In the end, it's still the residue water absorption, because water in IR, although you don't do the water resonance, but there's always some water absorption area, and that essentially limit. So we tried all the way to minimize the water background, because that absorption will produce noise. Yeah.
But if you could, in principle, go to, I mean, this for non-biological samples, to water-free environments, then you would be able to.
I think, yeah, in that case, I believe we can reach a single molecule detection.
Yeah. Okay.
Yeah, with a special solvent. Yeah.
Okay.
Yeah.
Is there anyone? So I have a question that, so when you use for living cell, and you are using quite high power,
yeah,
doesn't it have some kind of damaging effect in the cells, or what could be the limit for that?
For the, you mean the in SRS or NIR, in SRS, SRP? They eventually, yeah, it's a trade-off. So the idea, I think eventually the idea is to go to longer wavelengths. That's what we do here. We go to shorter wave IR. So use 1 micron and 1.3 micron, but there you can minimize the multi-photon absorption, right? So that can reduce the damage. And that's the beauty of stimulated Raman compared to spontaneous Raman, because you can go to longer wavelength to do this measurement. And I think that it's probably the future of the field is all go to longer wavelengths. Yeah.
Yeah. We have questions in the chat box. There is 2 questions.
Yeah.
From PC. So
how to distinguish SRP signal from other ultra-fast? Yeah, that's a very good question. It's yes, because as I mentioned that each of the beam can produce for example, 2-photon absorption process, and that is but that produces a linear background. And if you use longer wavelengths, you can minimize this 2-photon absorption. So how to do this is that you do the delay modulation. You delay the pump and Stokes pulse, and that will produce the SRP signal. But now the pulse is always there, so you don't see the linear absorption. That is a way that we do pump probe microscopy. But in general, that if you can, if you can do hyperspectral imaging, because the Raman signal is very sharp, so you can see the sharp peak from the background, and you can separate them in the spectral domain. That's another question is from, have you thought about using imaging for chemical quantification? Oh yeah, that's a very good question. Yeah, sure. I think quantitative imaging is very important. There are a lot of people doing this quantitative SR imaging. In SRP, it's you need to depends on the system, right? Because SRP depends on the environment as well. So you can so you have to know your system in order to do the quantitation. But in solution or in film, in simple sample, we have shown that we have beautiful linear relation with this. So we can in principle we can use calibration to do the quantitation.
Okay, thank you very much, Professor Cheng again.
Okay.